All right. So as you guys are settling in, I want to make a couple quick announcements. So this is one of our main heavy hitter speakers, one that a lot of you guys expressed interest in. This is one of our main topics this time is the use of core concepts. So we're going to be welcoming shortly Jenny McFarland. Jenny's going to be speaking and she'll have some interactive pieces in her talk. So we'll be running around with a microphone and getting some comments throughout. We'll also have some time specifically for Q&A with Jenny right after her presentation, followed by Claudia, who's going to talk about some of her work with how we might do some programmatic self-assessment for core concepts within our departments. And then we have quite a bit of time afterwards where we're really going to do a deep dive with some table discussions related to that. And after Claudia's talk is when we're going to ask you to shuffle around to some different tables. And these are really focused, really drill down concepts spinning off from some of the topics we've talked about earlier today. So that's what's coming. Talk from Jenny with some discussion, talk from Claudia, and then we'll do deep dive with some table discussions. And at that time, when we change, I'm going to give you a heads up and then I'll remind you again. We're going to ask that there's a person at each table who's going to be reporting and recording on the conversation that happens there. You'll be given a set of targeted questions at that time. So we are going to need someone at each table with access to a laptop and a plug. And we have a Google Doc that's already pre-made with the topic discussions. And Claudia's link is somewhere in your email. So when we do switch around, someone's going to be the responsible party for pulling up that document and gathering notes because we'll have a report out session and some of these topics will carry into tomorrow too. So that's kind of just a heads up on that. And again, as you're finishing settling in, the coffee and cookies will be over here. Help yourself at any time during this run here. Feel free to dismiss yourself as you need to find the restrooms again. So we really wanted to have Jenny come last year, and she really wanted to be there, but we planned the meeting right in the middle of her busy time of teaching, and she couldn't come. Yeah, and Joel's closer, and Joel came too, right? Joel is right next door. Yeah, so Jenny is definitely a wholehearted supporter of the work that we're doing, and vice versa. Jenny is one of our PMIG superheroes, I would say. Jenny is at Edmonds Community College, where she's served as the chair there of 11 of the last 18 years, and she's the only education tracked tenure stream faculty there, which is an amazing thing. And I would say one of my favorite things to talk about Jenny for, this is one way to give her the recognition that she deserves, is to know that she was the Claude Bernard Distinguished Lectureship for the APS last year for the teaching section, which is a huge, huge honor. And that comes from the work that she's done intellectually she's got a bunch of grant funding that we'll talk about and great publications and presentation histories. What she's going to talk about today is her work with core concepts. So her intellectual work and research work is related to conceptual understanding of core concepts and identifying those and unpacking those. And that's the stuff that we really want to hear about and really want to know about. She's also been the PI or co-PI on numerous NSF grants relating to the Leap Up program we heard earlier, Core Principles of Physiology, Physiology Educators Community of Practice. So she's really one of our thought leaders and spiritual leaders of our group here. She's got many, many publications and presentations, and she's going to speak to us a lot about one of her passions here, which is the Pulse community that we mentioned earlier, and those rubrics. She's been involved in departmental external evaluations and things like that. She has so much she can offer the group. It's just such an honor. And she will be talking, she also literally wrote the book that's up here. She'll show you about core concept with Joel. She's an author on the book of core concepts. And so as Jenny is talking, I want us to think collectively as we're moving into the discussions tomorrow and the work that Dee was just speaking about. When we're talking about core concepts of physiology, how does that relate to the service courses and intro courses that we're teaching in our department? How might those core concepts look different if you're teaching a majors versus a non-majors course? Do your majors take an introductory course and then spin off and take a different course? Are your majors in a totally different track? How does that look different? How do those courses look different? How did the HAPS objectives look differently when they're applied to those different courses that we're responsible for? And what does that look like if we say we want the core concepts to be the basis of a programmatic guidelines? What does that mean, and how does that look at the programmatic level if we write that up? What does that look like if they're really drilled down and unpacked? How does Jenny's core concept work tie with FISMAPs, tie with HAPS? And we're going to try to put our hats on together and sort of think about some of these issues. So kind of keep that in mind as Jenny is talking. So I'm incredibly grateful to have Jenny here. Thank you. Welcome. Take a seat. And I'll take them.
Yeah, my undergraduate training is in engineering, so it's showing now. So thanks so much for having me. We don't have physiology majors at community colleges, but it's super important for community colleges, as the two other people here from community colleges, for us to be part of your group. So I'm going to talk a little bit about the concept work we've done on core concepts and conceptual frameworks. I'm going to touch a little bit on assessments, but I'm really not going to talk about FISMAPs too much or the homeostasis concept inventory. We can talk about that later. There's a lot of stuff out there on that. I want to give a nod to our team. Joel Michael was here last year, Harold Mudell, Mary Pat is here, Bill Cliff, who's done a lot of work on case studies and other things, and the late Ann Wright. We're really integral in this work. It's a big team effort. The book is really due to Dee handing us and me saying no and Joel saying yes, so you can thank Dee and Joel for that, but we're very happy it's out there. Okay, so it's afternoon and I don't talk very much. I mean, I do talk, but I don't talk all the time. So this is a book out of the University of Washington, a study, qualitative study with lots of interviews. And I love this quote. It's one of their interview questions. So I'd like you to take a few seconds and think about your teaching over your years. What would you say are the big directions of change? So what's one big direction of change in your teaching, and what caused you to move in that direction? How is your teaching different now than when you started? I'd like you to think for about 15 seconds, turn to your neighbor, and share one idea with them. Okay, I'm going to stop people. I'm going to just call on a few people I know. So, Dee, have you changed your teaching over the last, decade or two? And how? So, in what directions has your teaching changed, and why did it change?
Well, I don't like standing up and talking, so I'd always had a very Socratic kind of class anyway. And I think the biggest change was actually, when I wrote a book, I went, I don't have to tell them all that stuff. It's in the book. I agree with everything that's in the book because I wrote it. So I'm going to give them reading assignments and expect them to have done it before class. And so I essentially flipped my classroom back around 1995.
Great. And do you want to pass the mic over to this table, just somewhere, just random? Anyone can. So how have you changed your teaching?
So I have been trying to impress upon the understanding of biological sciences totally from quantum mechanics point of view. So I am bringing consciously the quantum mechanical approach to biological sciences. Okay. For example, every biological process is an interplay of network of digital molecules and analog molecules. So the digital molecules are nothing but the nucleic acids, right? And the analog molecules are nothing but the various protein networks. And we see the fine modulation of this process by different carbohydrates and lipids. So this is a paradigm shift which I have been trying to bring in my teaching in India. So totally a quantum mechanical approach will be more interesting, and it will guide us for the future personalized medicine or predictive medicine or precision medicine.
So one of the things you've done is really find that students found it more interesting and increase student engagement. Is that part of what you're saying, that was more interesting problems, or am I misunderstanding?
No.
Anyway, so I just want to move on to one more person, but thank you for that, because I think flipping the classroom, other things,
the laws of nature which are applicable in physics and chemistry, they are also equally applicable in biological sciences.
Right, exactly.
The only issue is the operative aspects of the laws are very complex. So we need to take a stand to understand the biological process. We need to have a knowledge of higher mathematics and physics.
Yeah, exactly. So
the biological systems, though they are non-linear, they are very precise. So there is a lot of fidelity in the process. That is what is required for the future of precision medicine or personalized medicine.
Okay.
So my strong conviction, I have been telling to my students since more than 5 years in India, and how do we really bring the quantum mechanics to understand the biological sciences?
Okay, great. So usually I would spend a little bit more time on this, but I think this is really important to ask because I'm interested in departmental change. I'm also interested in individual change. And as we're looking at what we're doing in physiology departments, I think many of us come with the model that there are some recalcitrant people in our department, perhaps. But yet, if you start with this question, then everyone has something to offer. Most people aren't teaching exactly what they did in the way, and it's introspective, so I think that's important. I'm also, my cousin always tells me to come clean. I did play basketball in college and high school, but, I went to, I didn't go to a basketball powerhouse. And one of the things John Wooden said is, it's what you learn after you know it all that matters the most. And if there's anybody that knows it all, is people who teach physiology. Faculty are really in that position of thinking they know it all. And so he was looking at this in players and coaches when saying we must get our players to believe that the best way to improve the team is to improve themselves. And in doing so, we must not lose sight of the fact that the same principle holds true for the coach. So all of the things we're thinking of doing for our students, we also have to think of that change in terms of the faculty itself. So we really do have to think what's going on. So a question I often ask faculty is, what have you learned about learning in the last year? Because, really, it's what you learn after you know everything that you learn the most. So normally I would do another think, pair, share on this, because I think these two questions are really important to ask faculty as you're embarking on change. What has been the arc of your career, and what have you learned lately about learning? This is a slide that I an idea I got from a symposium I was part of in EB in 2016. This is a metaphor, of course. So education is not just a pile of facts, which would look like this, which are a bunch of stones up in Wales. This is the Galler's Oratory in Dingle. There's a difference between these two things, right? Science is built up of facts as a house is built up with stones, but a collection of facts is no more science than a heap of stones is a house. And I teach at a community college. There are many, many people who, coming from engineering, I think are just having their students memorize the names of widgets, hopefully with the dead guys' names associated to them. And to me, that isn't what I want to do. I want people to understand how things work, but we need a framework. We need to frame these facts for our students. And that's in part what we've hoped to do with the core concepts and conceptual frameworks. Facts are important. We need to have some facts, but we should choose the facts that we ask our students to know based on the end goal, which is conceptual understanding. So that in mind, this is a question we really do ask ourselves, I think, often. What do we want our students to know and be able to do? So this is, what are the curriculum guidelines for physiology departments? And what will they need to know in order to do this? And also, what won't they need to know? So is there anything that you currently teach, especially naming a widget, that you'd be willing to just let them have on their cell phone? So for me, I didn't take anatomy as undergrad because, I was busy taking differential equations. But when I thought the canal of Schlemm was really cool to say. But my students don't need to know that to solve problems, even in the anterior chamber of the eye. They don't need to know that. So what is it that you teach that they don't need to know in order to solve a problem? Is there anything you'd be willing to just let sit on their cell phone so they can look up that name or that? Yes? Golgi tendon organ. Hey, maybe this is the first transducer. Do we need to know that there's one more thing named after Golgi and not nearly enough named after Cajal? No. Who, after all, was correct. But I do think that, sorry, I keep pointing with the point of this, not here. If we need to be able to do this, because there are too many facts in there, so we need to be able to think about what is the scaffold, what processes are we asking them to understand, what concepts, what problems are we asking them to solve. Let's ask them to know the facts that help them solve that problem, and let's leave the other ones in Dee's fine textbook. Right, because they can look those up. Okay. What we have done in our group over the past, seems like a decade or so, is to help really leverage the community of people in physiology education to identify the core competency, the core concepts in physiology. And the medical school faculty in the Future Physicians Report talked about competencies. Vision and Changes talked about competencies. We've really focused on core concepts, and then we have unpacked those core concepts into a conceptual framework. What underlies this big idea of homeostasis, or this big idea of flux? We've also, along the way, identified misconceptions, again, with large numbers of faculty. We've described, but from the faculty perspective, we've described, Mary Pat and Jennifer Doherty are in the process of describing student learning progressions on some core concepts. We have a conceptual assessment of homeostasis, and then that allows us to align assessment with concepts. Today I'm going to talk a little bit about the core concepts in physiology, conceptual frameworks, and the learning progression work that's going on now. So what are core concepts? I think most of us know that, so I'm going to kind of skip over this slide. We owe a lot to Wiggins and McTighe in their enduring understandings, and to the work that led up to Vision and Change to really think about what do we want our students to know and be able to do 5 years, 10 years after they leave our classes. In our group, we've worked with the term core concepts and core principles. Harold Modell wrote the fabulous 2000 paper on general models. And again, we want to do this, keep pointing. We want to do this because using backwards design, we want our students to be able to solve problems with conceptual understanding and learn about what they need to know to solve those problems. And in order to do that, they need some conceptual understanding. So Vision and Change, there's lots of copies back there, came up with 5 core concepts. Not a lot of physiologists in the hundreds of people that led to that document. So their core concepts, although they're important in physiology, are missing some of the ways that physiologists would express what's important for our students to know. But certainly structure and function is very important. Information flow and storage. Information flow is super important in physiology, but I think most of us think about the neural and endocrine system and signaling, whereas most of biology are just going to consider the information from DNA. And then a lot of what physiologists care about is under this big category of systems. But clearly we care about structure and function. The Scientific Foundations for Future Physicians report came out with evolution and also homeostasis as 2 of the fundamental core concepts. But I can tell you when we surveyed faculty on these core concepts, so I'm flipping over to the physiology core concepts here. These are the 15 or so core concepts that Joel, Michael, and I got from about 90 physiology faculty from around the country and a few from outside the country. We basically did grounded theory. We sent out surveys. We got back narratives. We looked at them separately. We tried to group them. Then we came together and regrouped them. And then we sent them out to another broad group of physiologists and asked them to say whether or not they thought they were important and also rank order them. Surprisingly enough, there was one of these that many physiologists didn't think was important in their teaching of physiology or human anatomy and physiology. Anyone want to venture a guess as to which one it was? Evolution, the number 1 for most other biologists. And indeed, when you look at many of the textbooks, evolution is not really even mentioned. So that was surprising, but also interesting that was the one thing that many people disagreed on. Going backwards, looking at The earliest work that that really got me engaged in this work was Harold's paper in 2000 in Advances in Physiology Education, where he identified these general models. And cell-cell communication, homeostasis, flux, membranes are ones that came up again when we asked people to identify core concepts as a group of people. This is what you all said. So Claudia sent me some data, or somebody sent me data, about what you all said were important for your goals. And so you can't read it up here, but cell-cell communication is up here under the purple star, flow-down gradients or flux, homeostasis. These three are clearly important to you all and important to physiology writ large. And they're also three that we have unpacked and vetted the conceptual framework for, and those are all published actually in different articles in Advances in Physiology Education. Cell membrane is here, and mass balance, although I think if you come to Mary Pat's poster, we'll argue quite strongly that mass balance is super important. But you should have some little QR codes on your table. Just stick them in your name tag, and later tonight, before you get click on, put your stuff in up there and go to that. That will take you to our Physiology Concepts website, and it will take you to 2 abstracts from EB, 2 posters on unpacking these 2 core concepts. And under additional materials, there's a survey under each one. So if you want to be part of this process of unpacking core concepts, we are not doing this alone. We're doing it in community, and we want your feedback on the elements of the unpacking that has been done thus far. So if you can get that feedback and you can snail mail it, because Joel likes snail mail, you can print it out and mail it to Joel, or you can, whose address is there, or you can fill it out and email it to Joel. I keep telling him, that he cannot do the stone tablets anymore. But he won't let me put it on a Google form because he doesn't want to do it that way. But that's what we have. So we'd really, really, really appreciate it if you could help us out with that. Any questions about that? Have people seen the conceptual frameworks in the...
No, regarding feedback, activity, and anticipated behavior, another concept. Principles of optimization.
Oh, optimization? Yes. Optimization is really, I think, as an engineer, optimization is really important, but we're really looking at undergraduate physiology. And I think optimization is something that they're not really going to get to without computational skills that our students are getting. But I do think that optimization is really important, and that's important, in adaptation and other things. Yeah.
Biological sciences from a thermodynamic point of view, they are far from equilibrium. Inherently, they are unstable.
Right.
Instability is the basis for its growth and decay. To extend the phenomenon's growth and evolution,
Right.
different states of phase transitions, we have to bring the concept of feedback activity.
Right. And so part of what we did when we were coming up with the core concepts is we looked at input from a wide variety of physiologists, so not every suggested. So there were a few we're looking at feedback gains, optimization, and things were important, but this was really the consensus core concepts across a wide spectrum of physiologists. And we recognize that there are some things that some people are going to value and teach more than others. Okay. So this is the book. Thank you, Dee. APS members can get this, can download it for free. If you're buying the book, no one actually buys the hardcover. The money all goes to APS. So this is really an APS effort. We were urged to do it by the American Physiological Society, and it's been nice to see how the book has been used in the chapters. Has anybody downloaded the chapters and looked at those? And many of these chapters are derived from the papers, which already have a little bit more background in them. So when we looked at physiology core concepts from the faculty surveys, in the paper in 2011, which has the faculty survey results, that's where we did the unpacking of flux or flow-down gradients. Homeostasis unpacking was done, cell-cell communication was done just about a year ago, and Joel and Harold are working on the mass balance and cell membrane. But clearly, there's more left to do. So again, download it. I want to speak a lot about community because I think the narwhal example was really important, that we are a community. And this is work best done in community for a lot of reasons. People have really good ideas that I haven't considered. People think some of my ideas are really ill-considered. Joel and I have very different perspectives on things, as do other people I work with. Working with people that you have a different perspective from, I highly recommend. It makes your work so much better. But again, another John Wooden quote. Does everyone know who John Wooden was? Don't tell me you don't. No. He's a very famous basketball coach. But his advice was to find like-minded colleagues to form a team willing to work on improving classroom teaching and learning. And what we have done over many years, and many other people at HAPS and APS and the teaching section, is to work on fostering community, whether it's with the PCOP fellowships, with the now third biennial Institute on Teaching and Learning. And the work that we've done is deeply rooted in community. So when I look at our final report for our NSF grant was in 2005. In 2014, I just kind of pulled out our 3 project goals as they were stated in that report. One was to validate a conceptual framework for homeostasis. We did that. Construct and validate a concept inventory for homeostasis. But one of the most important things to me was to really foster a community of physiology faculty to do this work. Because pragmatically, if you don't do it, they're not going to use stuff they haven't been involved in developing. So that's one of the important kind of secrets of this group, is that we're developing stuff as a cohort. If a cohort gives input to guidelines, they're much more likely to use it than if they gave no input at all. And it makes your work stronger and better. So we, in NSF speak, the broader impact was really to help transform undergraduate physiology education and to support faculty who are employing concept-driven, evidence-based work that's going to move their students from naive understanding, this lower-level thinking, teleological thinking, to more closer and closer to expert thinking as they go through their programs. We recruited faculty back in the day from the HAPS listserv, from the APS teaching listserv, IUPS, a regional network in the Northwest. We've participated in PCOP, and I think we were just talking about writing a PCOP blog. How many people here have written a PCOP blog? Yeah, exactly. IPS ITL, SABRE. Anyone know about SABRE? Anyone not know about SABRE? Oh, Mary Pat, 30 seconds. SABRE?
Society for the Advancement of Biology Education Research. We have our next annual meeting in Minnesota, at the University of Minnesota.
Woohoo!
End of the month in July. And at this time, we have 330 people registered for the meeting. We have 60 talks on biology education research, 3 long talks, and we have 175 posters that we presented.
And we've slowly been growing physiology. Used to be, and still is probably a minority there, but we do need people presenting their work at SABRE because this is where we share stuff out. And physiology isn't as big enough a part of biology education research as it should be, because physiology is the most fun and the most important. So just, I don't know. So we've had somewhere between 200 and 300 faculty work with us, either in workshops where we make you give us data, or in talks or in poster sections where we nab you and make you give us data, or handing you various surveys where we make you give us data, or all kinds of ways. But it's really been the efforts of the community that have allowed us to do this work. So the question then comes back to, how can we help students both acquire factual knowledge and conceptual understanding? Since it's really difficult to get conceptual understanding with no facts whatsoever. The pile of facts approach really is, outside of Dee's textbook and a few others, the way textbooks are written. You march through the systems, you learn a bunch of stuff about them, you tow your students along, they listen to you talk about them. So this has been the traditional paradigm. We explain physiological function based on anatomical structures, system by system. Sound familiar? I never had an undergraduate physiology class, so I was not exposed to this. The paradigm that we're really proposing is to use core concepts and conceptual frameworks to scaffold conceptual understanding and then bring facts to bear that apply to those specific problems and those specific concepts. And then to really come back to the core concepts, even if you're going through it system by system, to come back and point out to students, hey, you already got this. This is just another, you're changing the resistance, so you're going to increase flow, or you're increasing the gradient or decreasing gradients. You've already learned this. We're now just looking, instead of blood flow, we're looking at airflow, or we're looking at ion flow. And similarly with homeostasis. So they need it scaffolded. They need it scaffolded like 20 times. Every time you see them, you need to come back and say, oh, here's the general model. Here's the core concept. So, back in 2000, not only did Harold write his paper, but this terrific publication came out, How People Learn. How many people are familiar with this one? It's still pretty darn terrific. And they mentioned conceptual frameworks in there twice, and there was no explanation of what they were, and no definition. So, what do we think they? So we were kind of like, what are these? I think I know what it means, but do we all have the same understanding? So what would a conceptual framework be? So talk to your neighbor for a minute. Okay, let me just grab, Claudia, what's a conceptual framework? Well, what did you guys talk about? You were talking about the meeting, because you're busy. Anybody here? Conceptual framework?
No, not really. we just said it was difficult to define.
It is. So I'm going to get somebody who hasn't talked yet. Andy, what did you guys talk about? I'll put you on the spot.
Jeff and I talked.
I know.
Can you hear me?
Yeah.
We talked about being kind of a spine or a core. I know in my classes I talk about a Christmas tree, and really what I'm trying to do is talk about the trunk, and we're going to hang all sorts of different ornaments on this thing, but don't get too attached to the glitter. I want to keep you on the trunk.
This is why I asked this question, because I've thought about this for 10 years, and that's a great metaphor. I'm going to send it over here, just for a second, because I'm picking up the phone. No. Go ahead.
They are foundational ideas that you realize the same principle applies across all the systems. It has different names and parts in each system, but it explains what's happening in each system. So it's very foundational. It is that trunk. It's the centerpiece.
Yeah, so... No, that's okay. So, because what I want to do, what I'm trying to do is just kind of model this, and one of the core principles that we work with when we go into departments is that we try to get everyone to talk once before someone talks twice, because I think what we want to do is hear from lots of voices. And the Christmas tree one is perfect, because students are going to resonate with that. That's a great metaphor. So, this is what we said it was. So, we think of it as a hierarchical structure for a core concept. How do we take a big idea and unpack it into smaller ideas? So in cell-cell communication, what is a sub-idea in cell-cell communication? What's something we really want our students to understand every time they see a cell communicating with another cell? Michelle? There's a signal, right? And if there's a signal, there's a receptor. So we want them, even at this basic level, we're going to unpack them. There's signals. There are receptors. There are half-lives or time spans. There's something that turns the signal off. There's usually a transduction process. So those are then separated out, and then they're divided into subcomponents, like, the signal could be, a protein, or it could be a steroid, or it could be, So it's hierarchical in that way. Unpacks a concept into constituent ideas. Those constituent ideas can then be, you can design activities around them, you can assess them, you can give feedback on them. It's pretty difficult to test whether or not students have an understanding of homeostasis writ large. That's a huge concept. So it organizes knowledge as the Christmas tree does. It organizes this knowledge. It gives them a scaffold to put it on. And it makes explicit tacit knowledge and assumptions. Because, I've been thinking about control systems dynamics for a very long time. My students have been thinking about them for 5 minutes. I have a lot of tacit knowledge that I don't even know I'm bringing to bear when I'm frustrated with, like, why don't they get this? But it helps make that tacit knowledge explicit. It builds connections to prior knowledge. Often, when we're talking about, a core concept like cell signaling, and we're talking about homeostasis, well, maybe there's receptors involved in both of those at some point, depending on the system. And it really does enable this development of a conceptual understanding by having pieces, having that kind of secret tacit knowledge and assumption that I hold, making sure I make it explicit to my students because I have this framework in front of me. Or at least, in my office that I think about before I'm designing things. So we've unpacked 3 of these. Flux, or flow-down gradients, 2011, homeostasis, 2016, cell-cell communication, 2017, mass balance, cell membrane, like 2017, 2018, we need your input to this. So, nothing would make Joel happier about my work than if I can say, 30 people just sent you surveys. I'm doing my part. Now you get to analyze them. So please help us. And the little QR codes are on your table, and that will get you there as well. So this is an example of a kind of just first and second level unpacking of the concept of flux. So we did this first because we thought it was easiest, just like Ohm's law. So flow is the movement of stuff, whatever that stuff is, from one place to another. It occurs because there's some type of gradient. And then we unpack the gradient. It could be thermal, it could be pressure, it could be partial pressures, it could be concentration. And then they can kind of get that, and then, by the way, more than one gradient can be present at a time. And that those 2 gradients now determine the magnitude and the direction of flow. So usually we have the osmotic gradient, hydrostatic gradients, or concentration and electrical gradients. But the magnitude of the flow is directly proportional to the magnitude of the gradient, or the balance of the magnitude of the gradients, and that resistance, and oftentimes a variable resistance, opposes flow. So there's kind of sub-unpacking under here. But these Roman numerals really are what students need at the bare minimum to understand this concept. And then we might want to throw some other more sophisticated things at them as they hang more ornaments on the little branches of the tree as we go through. So how can these frameworks be used in teaching and learning? So what do you all think? How could you possibly use a framework in teaching and learning? Yes?
Well, you had students say how Ohm's law is essentially the same thing as the Starling hypothesis.
Yes. Yeah, so, and we call it after these dead guys. So Ohm, Starling, Fick, what do they have in common? Right? No, exactly.
Yeah.
So at the level of student learning, we can use it at the level of student learning. And indeed, when we got cell-cell communication done several years ago, Joel revamped that unpacking for the endocrine system and gave it to his medical students to help them see those connections. So anything besides student learning, how else could you use it besides to directly affect your students' learning? Yes?
I use feedback control systems. That's on day 1.
Yeah.
Every single class, student.
Yeah.
In every single class, the student, whatever it is we're doing, start of the semester to the very end, they've got to identify what's the controller,
right.
Where's the receptor, what's the feedback, is it positive or negative feedback? In every single class they have to do that. So that's the one conceptual framework.
So one of the things we usually think about is scaffolding learning for students. and reminding that, guiding teaching and learning, so that that factual knowledge can be introduced to support the concept. Aligning assessments with the frameworks. It's like, oh, do I have, 20 questions that are just, in the case of homeostasis, on the effectors, and I haven't talked about the sensor at all? It can help you look through and really align your activities with the framework, your assessments with the framework. So for example, make sure that, you're talking about more than one gradient in your assessment if you work through a formative assessment on it. At the level of departments, however, they can be used in course design. They can be used to inform guidelines here, but curriculum mapping throughout the curriculum. And one of the things we saw when we were unpacking cell, and we took it to EB and we took it to other places, is everyone was saying that somebody else covered signal termination. It's like, oh no, they did that in biochem. Oh no, I think they do it over here. And it's like, oh, maybe you should check and make sure, because students don't score very well on that, and maybe it's because we all thought it's over there. Instructors can help to reveal connections between systems, between concepts, between ideas. It can, again, uncover assumptions and tacit knowledge. We can use it for alignment. And students can really use it to construct a mental model, or at very least this hierarchy, this Christmas tree that they're hanging it on, to scaffold understanding, especially as they go from 200 level classes to 300 level classes on to medical school. Yes?
So at the top in the curriculum mapping of concepts, one of the things that I've seen, many of us have seen this, is that people will talk about the same concepts in different courses, but use different terminologies and say, oh, by the way, this is the same as that you'll see in a different place. So is there some way that we could make some more common vocabulary so that they realize they're learning the same thing, just in maybe a new language or a new terminology or whatever, new system?
No, which brings us to Harold's paper in 2005, where they recommended some diagrams and we really panned some others. The consistent definition of terms, or at least the instructor knowing what terms are being used, and understanding what variables are being regulated. So this is just to think about the products. We have a lovely paper out, authored by Harold, on the concept of homeostasis. We have the framework. We have a concept inventory. We have aggregated misconceptions that's not published yet.
Suppose if you replace homeostasis with frontal pattern generators, that is more meaning than homeostasis. And also, it involves the evolution aspect of frontal pattern generators.
Yes. No, yeah, and I think that's an important point, but I think, again, we're trying to use a term that is used by that most physiologists have agreed to use. So, although it is somewhat problematic. So we have this kind of tear-out table of terms. We have a tear-out table of variables. It's too small to see, but we realize that people use different terms, and that's very confusing for our initial students. And then we have, what are the regulated variables we want undergraduates to know about? So that's what's in here. And then we think we should ask the same questions in every system. What's the regulated variable in the system? Where's the sensor? What might be some effectors? Is there a control center? Is it always in the nervous system? That's a misconception I think we give them, and then it's very hard to displace. So if you're asking the same set of questions about your Christmas tree, they're decorating the tree in every system, and then you ask the same questions, then hopefully you'll reinforce the fact that this is the same phenomenon. And am I out of time? Is that why you're saying? No. 5 minutes. There are some important sticky points that the conceptual framework allowed us to really get to. Students, not only do they fail to generalize, but they overgeneralize, to think that all negative feedback systems are homeostatic. Well, stretch reflex is homeostatic, but it's negative feedback. It's really not homeostatic in the way we think of it. Some of them think it acts like an on-off switch. Some students confuse effector with the response. I want to get to another topic, and I'm not going to have time to get to some of what I want to talk about. What I do want to talk about, this program out of the Pulse effort, which is aligning vision and change. I think I have enough here for every department. And this, in the last 5 years, we've come up with a few products for departments. One is departmental rubrics to help you assess where your department is in aligning with vision and change, both the core concepts and the recommendations around active learning. The Pulse Ambassador Program is one that we have 4 more years of funding for, that will send 2 to 5 trained facilitators as a team to your department, depending on the size of your department, to foster dialogue around teaching and learning, the Vision Tree department, and change efforts. Because we realize, just like in my department, no one listens to me because, I've been there for a long time and they know what I'm going to say. But if I go to a different department and say exactly the same thing that somebody else has been saying, they're like, whoa, that's a great idea. And they also play well with others when somebody from the outside is there. The little tensions sometimes aren't there. But we can then get them to think about goals, get them to think about how tensions sometimes are synergistic and differences of opinion, like between Joel and I, for example, can really lead to positive change. So that's the handout that's coming around. There's also a website here. The goals really are to engage each department in critical dialogue around vision and change recommendations, but this really is maps on to what you guys are doing. We'd love to come to some more physiology departments to engage in really real goal setting where when we leave, they have at least some low-hanging fruit goals and some long-term goals where they can say, Joe's going to be in charge of this, and Andy's going to be in charge of this, and Nancy's going to be in charge of this, and here's a deadline for going and talking to the provost about funding or doing this or that. We also meet with your deans or your provost or your chairs, your vice president or whoever's in the hierarchy, because if we do the ask and say, oh, these people are doing this great thing, they're thinking of asking for X, Y, and Z, they're not going to tell us no. They might, at worst, they'll say, well, think about it. But when the faculty come and ask, sometimes it's perceived as our faculty think they're going to just think that we're whining about more money. So we can help leverage that. It's usually 2.5 days. So we usually meet with the organizing team the night before, have 2 full days of workshops. The travel and the workshop costs are funded by a grant. Depending on the resources of the department, we sometimes ask for food and coffee to be provided by the department. Lodging is sometimes provided by the grant and sometimes provided by the department. But it's a great effort. Those short-term goals are really to get departments to come together, use empathic listening and some leadership skills, identify barriers. And I think for people who are leaders in here, most faculty like to say the barrier is one step up. Most barriers for change are owned by faculty. And you need to have faculty acknowledge that so they can move forward and develop an action plan. The long-term goals are really to help the department over the next few years by just being aware of resources that are out there, Pulse resources, but also other resources in the physiology world, to try to buy in some institutional commitment and faculty commitment. How much, do I have 2 minutes left? Yes.
This time is just time for your question and answer.
Okay.
We have actually 15 minutes before flying stop. So
Okay. Okay, I want to go briefly through, so this is Dr. Doherty, Dr. Wenderoth, who are with Jack and Emily and a bunch of other people at Michigan State, who are working on looking at, from a very student-centered approach, looking at students' responses to interviews and questions around 2 concepts, mass balance and flux. The work that we've done up to now in our group has been really faculty-focused. What do you think your student misconceptions are? What do you think that the expert-level conceptual framework would be? And this is really kind of ground-truthing it with learning progressions that are based on student responses. So the learning progressions really are a successively more sophisticated way of thinking about a topic. It's really embarrassing talking about this around Mary Pat, who knows more about than I do. They're anchored with naive student reasoning on one end and expert understanding on the other. So this is, they're going from the upper anchor to the lower anchor. So What do naive students do that just coming in? What do your intermediate, sophomore level students think? Your upper anchor, your graduate students or exiting seniors. And this is where the conceptual framework derived by physiology faculty can be kind of a nice guideline. It's like, oh, this is where the upper anchor is. A long-term goal is development of assessment tools and teaching strategies. So this year, we're getting student data from Michigan State, UW, Edmonds Community College. There's about 4,000 or 5,000 colleges and universities in the United States. We'll be asking people for help as we recruit, especially get the machine learning to work and get more, because we want to make sure that the student understanding is representative of students across the country. We don't have, Michigan State, UW, and Edmonds are not Hispanic serving colleges. You know, they're not, just did something bad. So I'm going to let Mary Pat go through these, but really developing the learning progression framework, we're going to talk about on the poster. The thing that we're aiming for here, we writ large, is to send these student responses and to train the machine learning at the automated analysis of construction response group at Michigan State, to take in the student data, code it, score it, expert score it, send it into the machine model, get a predicted score, come back, iterate with new data till the machine score is similar to the expert score. And when we get there, then we'll be able to analyze massive amounts of data. So I skipped over some stuff, but what I do want to really plug here there are some work in progress. And we hope to hear about more work as we go on today, but the work done in physiology education needs the work of the community. So the 2 new conceptual frameworks, Joel is working on cell membrane and Harold on mass balance. Please, please, please fill out those surveys and get back to us. When you do hear from the Leap Up group in the next year or two, as we're trying to recruit new students, please respond, because we don't know exactly how that reach out is going to work, but you know, it's going to be important to get more. And then the Pulse Ambassador Program is out there as a way to come to your departments and help you work within your departments and try to get people on board and come up with the shared vision for your department. So I want to leave you in part with this slide that, again, we can't do this work without like-minded colleagues, and that's why it's so wonderful for me to be here, such a lovely invitation from Erica, Claudia, and things to be here, because, you know, these are our people. And our people exist in the teaching section and at HAPS and a few other places. But this is where we can go and get that support and, you know, nourish the young and protect them as they come up. But we do need this fellowship of folk that are transforming teaching and learning in physiology. So thank you for your attention, and I'm happy to take questions. And again, we really want to acknowledge everyone in our group. We have a wonderful statistician who's now back in Prague that helps us with this. The APS teaching section at HAPS, we could not have done this work without them. Hundreds and hundreds and hundreds of students who have written responses and answered surveys, and the NSF and the work of Pulse. So that is what I had to say. Yes?
Do you do the pulse visits above the departmental level, so pulling in, I mean, life sciences, right, that could be biology department, chemistry department, a physiology department. Have you done any of those ambassador visits with multiple departments?
We have just done a visit at a well-known Midwest private liberal arts college for all of science. In some community colleges, we've done it for all of STEM. Because apart from the core concepts, everything else is the same. I'm on an advisory board for a geosciences grant on transforming geoscience departments, and it's way more fun than talking to A&P faculty because I don't have any baggage in geology. But the only thing that's different are their core concepts. And so, yes, we have done it depending on the organizational unit. We're pretty flexible. The one thing we do want is to get to really have you try to get 90 to 100% of the faculty there, but certainly over half the faculty. And that includes bringing adjuncts in, because for many of us that's important. And we also require that we do have a meeting with your dean or VP or whoever is above that, because we really think that we can provide some leverage that's different than the leverage you can apply.
So I'll just make a quick comment about the value of these programs and give a plug for Jen's poster tomorrow, Jen Rogers. So we did some pre and post surveys at the PMIG meeting last year, and then we did like a 6-month follow-up survey, and a lot of people said, oh my gosh, I had great ideas, this is so great, and we have the data that says like how easy was that for you to go back to your department and implement. So Jen has some data on a poster about that tomorrow, but this kind of work here is really where like the rubber hits the road, right? So like you have great ideas, but like how do you get your department to work with you? Like how do you actually make this happen, and how do you have an external voice besides you just coming back being like, yay, this is so exciting, everybody, and they're like, yeah. But having your department invest in something like this, or having people come out, is really, really important, because I think there is a huge barrier to actually big-scale, large-scale implementation and change like that.
And we've really been trying to get the facilitators trained across the country so people don't, first of all, that decreases the travel costs, but mostly it means that there's somebody in your neck of the woods who you can touch base with later after the visit. And there are follow-ups and there are some other things. But I think that getting change to happen and adopting curriculum changes, adopting guidelines, we've learned a lot about shared ownership from the work that we've done, and shared ownership is so important at the departmental level. And if someone tenured feels excluded, it can do, as we all know, it can really derail that. So how do you build that and include those people?
And then Jenny, what about training new ambassadors? So like if we want to sort of support each other in physiology in this group, right? Like how are you training new ambassadors, new facilitators?
Well, I just spent 3.5 days doing that last week. So there was just an ambassador training program last week at University of Puget Sound, and there'll be another one next year in Georgia, and then I can't remember where the next one is going to be, but we're really trying to grow this, and I think one of the things that we do want to do is add some more physiology faculty, although you're going into life science, and so we usually say we're going into life science departments, but again, we've done all of STEM, we've done all of science, but really it's and a lot of other scientists haven't read Vision and Change, right? Most biologists haven't read Vision and Change. But that idea of transforming departments is something that's happening all over in science, and it sometimes is easier to talk to people outside your discipline than in it, too. But it's certainly easier to talk to people that aren't at your home institution.
And then I just want to give a plug, like Mary Pat, Jenny, like when you guys have these projects, when others have projects, so we're collecting a listserv, right, for this organization. And I know, like we're saying, all of us are like, oh, we're advertising through the SABRE listserv, through the APS teaching section network. Like, just keep in mind, like, we want to have a two-way communication, like on our discussion board too. Like, this is a great way to make connections and get more people. So if Mary Pat, like, if you want to send out something through our listserv, like, please do. And that will help us.
So the idea of using the core concepts as a framework to scaffold conceptual understanding, instead of doing a system-by-system approach. And I've been thinking about what that would really look like.
It doesn't have to be instead of a system-by-system approach, but I think it has to be instead of driving it with content. It's like, here, let's steam 100 widgets, and then let's talk about homeostasis. It's like, well, why don't we move into this new system? Say we've just done, neuro and talked about sodium-potassium channels and, Nernst equation, and now we're in cardiovascular. And say, well, where is flux important here? How is the movement of blood like the movement of ions? Where is the variable resistance? Where is the gradient? What kind of gradient is there? Oh, look, in capillary exchange, there's 2 gradients again. Gee, that was really hard in the Nernst equation, but now we can leverage that model that we got before. Yeah, it's pressure gradients and osmotic instead of concentration and electrical, but we're reinforcing that general principle. And then maybe we have to leave some facts in the textbook and on the cell phone so that we have time to really invest in, oh, we've seen this model before, let's work a problem that we can use a scaffold from the previous, the example group activity we did 2 weeks ago. So you can still teach system by system. The problem is, are we content-centered in naming things and memorizing stuff, or are we process and concept-centered in saying, oh, I know I'm going to talk about this, but maybe I'm not going to talk about this part of this chapter at all because I'm going to spend time reinforcing this core concept and really solidify it as opposed to just running through the entire chapter.
So my question is, to me, the hardest thing is what to leave out. And Nancy and I were talking about this at dinner last night. We were just saying how if this is their only exposure to physiology, this is the only class they're going to take, they're not necessarily a physiology major, they might just go to nursing and that's it, that's all they're getting. You feel a sense of responsibility, and so that decision is a tough one.
But I think we have to, it's the illusion of coverage. So we're wedded to what? So if I'm behind, I just talk louder and faster, and then they learn more. And it's like, well, they're not learning it, dude. If you just have them memorize stuff and dump it and measure what they memorized, low-level blooms, 2 months later, so it doesn't matter. And this is what I really love about this book. The title of this book. It's the illusion of coverage. They haven't learned it long term. It's not these names of 1,000 widgets are not an enduring understanding. So we're wedded to an illusion that if I've covered it, I check off I've taught it, they haven't learned it long term, but my job is done. Well, I think my job is done when they've actually learned it long term. And so I think that we're operating under that illusion. So when you realize it's an illusion that that we have to have them memorize these 1,000 things, and if we don't do it, they're not going to be prepared for nursing. But let me tell you, when they enter their nursing program, that knowledge is decayed. So, I'm really glad that I spent 30 years doing this.
So what is it that they're going to remember 2-3 years from now? What is it that if you ask them, you run into them down the street and you ask something maybe.
They're going to remember, because,
If something is moving, I got to look for a gradient.
Yeah.
I got to look for resistance. I don't know what the gradient is, but there's got to be one there.
And it's like with muscles. I don't care if they remember anything about a particular muscle, but I want them to know when they go back, it's like, oh, I need to know 4 things about this muscle. I need to know origin, insertion, action, innervation, and that's what I know I need to, that's my framework that's what I need to go learn, because I know how to learn it now, and I've forgotten every muscle I've ever learned, but at least I know what I need to learn. Mm-hmm. And it's that, and it comes from my, engineering background, but you do remember for decades processes and basic core concepts of mechanistic thinking, and that's what we want them to know, because. And people say, oh, well, they're just going to be a nurse. It's like, well, in a trauma unit, the nurse is spending like 23 hours a day with you, and the doctor's spending 5 minutes. So the nurse needs to understand mechanistic thinking. So, but that is what endures. Yes, Michelle.
So I like practical examples. Can you give me, Jenny, a practical example of how you've done the core concept, any core concept, throughout a physiology intro course?
Let me see if this will work very well, but I'm going to just walk through some of these slides because I think this is, so when we're looking at the learning progression here, students usually start by storytelling. So if I'm teaching muscle contraction, they're going to learn a story from acetylcholine release to, actin myosin. And then if I perturb that by giving a toxin or something, they want to just tell me the story again. It's like, well, the story's changed now. But they want to tell me their story. We want them to start building something like this. So if we, for example, give them, this is a question from Mary Pat and Jennifer's work, on membrane potential, when you're looking at this figure, we have this net movement of potassium ions out of the cell. What can we change to cause net movement into the cell? So if and I don't do this, but if I was doing this and they'd work through these problems and learned about this, and I'm like, oh, we have blood flow. What can I change to increase flow rate? Or respiratory, asthma. It's like, what changed? So now I'm asking a different, not what kind of change, but what changed in this person that now flow, exhalation is taking so much longer? Was it the gradient that changed? In this case, no, it was the resistance. So we can have them explore these different elements of flux by coming back to those elements, gradients, resistance, and flow. And because, this was such a nice, elegant question. And generate as many ways as you can for how you can get potassium to move into the cell. Or how many ways can you think of to get blood flow to change? So I think that's a really important way of coming back all the time and saying, remember, we're thinking this is now a flux problem. What do you know you have to look for? You have to look for resistances. You have to look for gradients. What possible gradients could there be? And that's one of the things I say. It's like, I turn my class, so what gradients do you think we'll look for? Talk to your neighbor. Let's throw out a few. Some of them might be wrong, but that's okay. So is that helpful?
Can I actually get a follow up on that too? So Patrick has a poster on this. Like he has actually redesigned a course completely around core concept. Well, but I mean, I'm giving you lots of credit. But we're also going to have one of our discussion tables after Claudia's talk is going to have Patrick at the table and specifically talking about how are we teaching this? That's one of our discussion topics. So hopefully we have a lot of people that are interested in that and want to keep talking about it.
Right. And I think there are a number of us in the room that are thinking along these lines. Mary Pat does this a lot. Anybody else because we do, it's like, how do we use this concept again? How do we use homeostasis again? And I talk to my students, it's like, if homeostasis comes in wearing an overcoat, we think it's a new person, right? It's like, no, it's still a homeostatic mechanism. How do we get them to recognize that? And I think one of the ways we get them to recognize that is, in the Herald paper, we had these, every time I want to ask the same questions, exactly the same questions, like what is a regulated variable? Where is the sensor? Where is the control center? And if I do that, if I have this patterned set of questions and I have a standardized worksheet, it's like, okay, they're going to answer, then they really do get that exercise of familiarity. Oh, I do know this.
Any other questions? Okay, thank you very much, Jenny.
So if I haven't mentioned that, Harold and Joel would like to hear from you.